Hello, everyone, and welcome to today's webinar titled Continuous Bioprocessing, New Challenges and Opportunities for Membrane Technology. This webinar is part of the fourth annual Bioprocessing virtual event. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I'll now present today's speaker, Andrew L. Zydney, PhD, Kunkle Chair and Professor of Chemical Engineering, Director, Membrane Science, Engineering and Technology, MAST Center, the Pennsylvania State University. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dr. Zydney, you may now begin your presentation.
Well, thank you for the introduction. It's really my pleasure to be able to be here today to tell you about some of the work that my group has been doing in the area of continuous bioprocessing, with a particular focus on the application of membrane technology for the continuous purification of monoclonal antibody products. If you look at the current portfolio of drugs that are provided by the biopharmaceutical industry, you see that the bestseller list is dominated by monoclonal antibody and monoclonal antibody related products like fusion proteins. Annual sales of monoclonal antibodies are now greater than $100 billion a year. And there are exciting new opportunities. Some of you may have studied the press reports on the release of monoclonal antibodies for the treatment of Alzheimer's, as well as COVID-19. These open up exciting new avenues for the development of biotherapeutics. Now, if you look at the overview of the manufacturing process for producing monoclonal antibodies, they all look generally the same, beginning with a bioreactor, either single use or stainless steel. In the case of antibodies, typically used to grow Chinese hamster ovary cells. And then you go through a series of steps involving clarification, primary capture, polishing, and formulation. Now, the industry today accomplishes this purification or set of operations in a fully batch mode. That is, each step in the process is completed before the next step is even begun. So you would grow your cells and they would produce your product in the bioreactor. At the end of that process, you would clarify the entire suspension to remove host cells and cell debris. You would then take that entire batch of material and go through the primary capture step and then subsequently through polishing and formulation. This typically requires large stainless steel tanks. The standard bioreactor for monoclonal antibody production is usually a 20,000 liter size reactor. And it requires a long period of time. Typically, if you look at the time going from the beginning of the process to actually having a formulated drug product, it averages about 100 days. Now, this type of production process has been recognized for many years as having a number of shortcomings. This is a quote from Robert Bradway, the CEO of Amgen, in an article that was published in the MIT Technology Review just about a decade ago. And he specifically comments that today's systems are hugely expensive and can take months. And with more efficient processes in place, companies could not only increase production of drugs in high demand, but they could produce medicines for rare diseases more cost effectively. Now, this is particularly true or important for some of the newer applications of monoclonal antibodies. As I mentioned, the FDA granted accelerated approval for Biogen's therapeutic antibody for the treatment of Alzheimer's just this past summer. It's a monoclonal antibody that has been shown to reduce amyloid beta plaques. The cost when it was introduced was $56,000 per patient per year. If you look at the number of people in the United States alone who currently live with Alzheimer's, this has a total market potential of $350 billion a year, which is more than 40% of the total Medicare expenditures in the United States. This is clearly not sustainable, and we've seen the additional challenges on the healthcare system that have occurred because of COVID-19 over the past 2 years. Something needs to be done to reduce the cost of production of these types of therapeutics if they are ultimately going to achieve the clinical benefits that we would ideally like to see. Now, there are a number of strategies that can be followed to improve the cost of manufacturing, but one of these that has been identified specifically by the FDA is continuous manufacturing. As Janet Woodcock specifically noted, she was at the time Associate Director of the FDA, there have been very little change in manufacturing platforms, and it was her hypothesis back in 2011 that there would, over the next 25 years, be an abandonment of those practices in favor of cleaner, more flexible, and more efficient continuous manufacturing processes. Now, why would one be interested in continuous bioprocessing? Well, if you think about the switch from batch to continuous processing, you can eliminate the hold tanks that are required in current batch manufacturing processes if your product moves continuously from one unit operation to another. In addition, because all of the operations are performed at the same time, the size of the manufacturing equipment shrinks dramatically for a continuous process relative to that for a batch process. And thus, the overall capital cost for a continuous process can be orders of magnitude smaller than that for a batch process. In addition, we can get much greater productivity because all of the equipment is in use at all times. We don't have to wait for one operation to finish before the next operation starts. And we can eliminate all of the non-value added steps that are often required in bioprocessing of batch systems. For example, the cleaning and validation before and after each step. There can be greater flexibility for continuous processing systems, allowing them to be used in multi-product systems where we can have closed systems with minimal transfers and better utilization of the manufacturing suite. And there's even the potential for improved product quality. Typically, products that are produced in a continuous system will have much shorter residence times within the process, which leads to less aggregation and less product degradation. And there's a more uniform microenvironment, which leads to better control over things like glycosylation patterns and extent of deamidation. Now, my group has been very actively involved in looking at opportunities for the development of continuous bioprocessing systems. And I wanted to take the time I had today to go over 3 short examples. The first is on the final formulation step by diafiltration. The second is a continuous countercurrent tangential chromatography that could be used in place of column chromatography for the purification of monoclonal antibodies. And finally, a precipitation washing procedure that could be a low-cost alternative to protein A for initial monoclonal antibody capture. So let me begin with the final formulation step and then work my way backwards in the process. Today, formulation of monoclonal antibodies, and in fact, most protein therapeutics, is performed using batch diafiltration. In this case, our product stream is contained in a recycle tank in the system and is continuously recycled through a membrane unit that has very high retention for the monoclonal antibody, but lets both buffer components as well as water pass through the membrane. In this case, we continually add in a diafiltration buffer and wash away the previous buffer components to formulate our monoclonal antibody product in an appropriate buffer for storage and delivery to the patient. Now, this is an inherently batch process. The new buffer, which I call the diafiltration buffer, is slowly added into the product solution while the old buffer components are filtered out through the membrane module. If you think about this way the process runs, our product solution begins in the penultimate buffer, and it is slowly replaced with the new formulation buffer, and it doesn't get fully into that formulation buffer until the very end of the process. In addition, the product needs to be recycled multiple times through the ultrafiltration module, subjecting the monoclonal antibody to multiple pump passes, which could lead to degradation or aggregation and loss of quality in our antibody product. Now, how might we consider switching this to a continuous process? Well, in its simplest form, diafiltration is nothing more than a dilution combined with a concentration step. In the batch diafiltration process, we do that dilution and concentration simultaneously, so the volume of our product solution doesn't change with time. But in a continuous process, we could think about doing this sequentially, where we would take our antibody feed, dilute it with a buffer, and then reconcentrate it in a membrane module, which would remove the old buffer components and leave our antibody in the desired new buffer. Now, although this works in principle, it does not work in practice, because in order to get 1,000-fold reduction in the concentration of any of the buffer components that are currently present with the monoclonal antibody product, you would need to use a diafiltration buffer flow rate that was 999 times larger than the flow rate of your antibody product. That's completely impractical. First, it would require way more buffer than one could ever utilize as part of a purification process. And secondly, reconcentrating the antibody by a factor of 1,000 in a single pass through a membrane module is currently well beyond the technology of even the best membrane modules that are available commercially. So instead, what we have decided to do is to look at a countercurrent stage diafiltration process that accomplishes the same thing. That is, we do our diafiltration continuously in a single pass through the membrane unit, but it functions in a practical way with relatively low flow rates of buffer and relatively low requirements for the concentration step by taking advantage of countercurrent staging. Now, this is a diagram of the process. In order to see the countercurrent staging, it's helpful to look at the flow rate of the diafiltration buffer and the monoclonal antibody separately. So the diafiltration buffer is added to the first module on the left, and we then take the permeate from that module, put it into the second module, and then into the third module. And thus, if we were to follow the flow of the diafiltration buffer, it goes from left to right from module 1, 2 to 3. In contrast, if we look at the monoclonal antibody, it's actually added into the third module, the one that's furthest to the right, and the retentate from that module is cycled back to the second module, and then the retentate that exits the second module is cycled back to the first module. Thus, the antibody is moving from right to left, countercurrent to the direction of the diafiltration buffer. This allows us to collect the purified or diafiltered antibody product out of the very first module, right where it is contacted with the diafiltration buffer. Each of the modules allows us to remove some of the penultimate buffer components, eventually getting to the desired degree of diafiltration, but we effectively reuse the buffer so that our overall buffer requirements are kept quite low. For those of you with a chemical engineering background, this is in many ways analogous to the operation of mixer settler systems for staged extraction, where the settler is being replaced by the membrane module, and the mixing actually occurs in small static mixers that we use to mix the diafiltration buffer and the monoclonal antibody product right at the exit to each of the membrane modules. The countercurrent staging, as I mentioned, allows us to get high performance with relatively little buffer flow in the system. So we used a model system to demonstrate the capability of this device. In this case, we used vitamin B12 as a model impurity because it's very easy to detect by a colorimetric assay. We performed our diafiltration with an IgG or antibody feed at a concentration of 120 grams per liter that contained 2,800 milligrams per liter of vitamin B12. And these are the results for the vitamin B12 concentration in the diafiltered product that leaves this three-stage diafiltration process. As you can see, the concentration of vitamin B12 is perfectly stable over a 24-hour process. We ran this continuously without any cleaning or regeneration of the membrane module. The vitamin B12 leaving this system is about 1,000-fold smaller than the vitamin B12 concentration in the feed. That is, we're removing 99.9% of the vitamin B12 in a single pass through the three-stage system. And this actually requires about 30% less buffer than traditional batch diafiltration processes for formulation of a monoclonal antibody product. Now, we can actually look at the optimization of this type of countercurrent stage diafiltration. It's really a trade-off between the number of stages and the amount of diafiltration buffer that we use in the process. And what I've shown here is the predicted values of the fold removal, that is, how much of an impurity can we remove, as a function of the flow rate ratio on the x-axis for a single-stage system, n equals 1, a two-stage system, n equals 2, and then a 3 and a 4-stage system. A typical target in a bioprocess might be 1,000-fold removal of an impurity that was present in the system or a buffer component. As you can see, we can do this in a 3-stage system using a flow rate ratio of just about 10. If we tried to do this in a 2-stage system, we would require about a 35-fold flow rate ratio, or a factor of 3.5 more buffer. And if we tried to do it in a single-stage system, the buffer requirements would be so large that they're well off this axis. The symbols on this plot are experimental data. We had both a 2-stage and a 3-stage system that we built in our lab. And you can see that the data are in excellent agreement with the model calculations, allowing us to use this type of analysis to optimize the design of a countercurrent stage diafiltration process based on the cost of the membrane modules, which is proportional to the number of stages, the cost of the buffer, which is, of course, proportional to the buffer flow rate, and the targeted degree of impurity removal. Now I want to switch gears and talk about another example, countercurrent tangential chromatography. Most of you are probably familiar with protein A chromatography, which is used for the purification of essentially all monoclonal antibody products. In this case, one takes a protein A ligand, covalently links it to the pore structure of a chromatography resin, and then packs that into a column. You can then pump your antibody solution through that column, bind the antibody to the protein A ligand, wash away impurities, change the buffer conditions to elute the product, strip away any residual impurities, and then regenerate the column for a subsequent cycle. Again, this is an inherently batch process. You can't start the washing step until you've bound all your product. You can't start the elution step until you've washed away all the impurities. Now, there have been a number of attempts to make what I would consider to be pseudo-continuous chromatography processes using multiple columns that are configured so that you switch between one column and another. I'm not going to discuss those, but I'm going to instead focus on an alternative approach, which we have referred to as tangential flow chromatography. In this case, we take the chromatography resin, which are these large bluish particles. We use protein A ligands that are bound to those resins, but now instead of packing the resin in a column, we actually use it in the form of a flowing slurry. And we flow that slurry over a membrane that allows us to filter out the host cell proteins while retaining the large resin particles that have bound the monoclonal antibody product. In principle, we could use the exact same approach for recovering the antibody. In this case, we would switch the buffer. The antibody would come off of the chromatography resin particles. It could be filtered through the membrane, and we would recover the purified and eluted antibody product in the permeate, while the resin particles are retained by the membrane and recycled back in the system. Thus, this new process of continuous countercurrent tangential chromatography, or CCTC, exploits the benefits of affinity binding in a chromatography step, but it uses countercurrent staging to improve the performance of the binding, washing, and elution steps to reduce the amount of buffer and enhance the yield of our antibody product. All of the operations are performed on this flowing slurry, and we use a combination of hollow fiber membrane modules and static mixers for the fluid and resin management, that is, to give sufficient residence time for binding, and then to be able to remove host cell proteins in the washing step and our monoclonal antibody product in the elution step. This really functions as essentially a conveyor belt. Our resin particles go through the binding step, then the wash step, then the elution step, stripping and equilibration. Here we've shown actually two wash steps at different conductivities. The resins just cycle continuously through and then back to the beginning. Meanwhile, in the binding and wash steps, any host cell proteins that go through the membrane are collected as waste. In the elution step, we of course collect our purified product, where that product could then be immediately taken to subsequent steps in the downstream process. This is truly continuous and steady state. All the resin is in use at all times, with some of the resin in the binding step and some in wash, some in elution, and some in stripping and equilibration. And it's truly at steady state. That is, we continuously pull off product in the elution step at a constant flow rate and a constant product concentration. Now, as I mentioned, each stage in the chromatography system is really just a combination of a static mixer and a hollow fiber membrane module. The static mixer provides the residence time for binding and desorption. The hollow fiber membrane module allows us to completely separate the resin from the fluid phase, allowing us to recover the resin particles and wash away all of the host cell proteins. This is a typical diagram of our picture of what this system might look like. We have a series of these hollow fiber membrane modules and static mixers. They're put in a series arrangement so that we can do countercurrent staging in a way that is very similar to the staging that I talked about in the diafiltration process. That's shown here in a two-stage system where each stage has a static mixer and a hollow fiber membrane module. The resin slurry comes into this system on the left, goes through the static mixer, the hollow fiber membrane module, it's retained, and then it goes into stage 2. In contrast, in this case for the elution step, the elution buffer is added into stage 2, and the permeate from stage 2 is recycled back to stage 1, and that allows us to collect the product from that stage. The staging is absolutely essential to getting high yield of the product. This is a comparison of experimental data and model calculations for the performance of the elution step using a flow rate of the elution buffer to the slurry flow rate of 3. If we did this in a single stage system, we could only recover about 77% of the product because too much of the product would stay with the flowing slurry. As we go up to 2 stages, we can get to 94%. 3 stages, we can get to 98%. And you could even drive this higher if you were to add more stages. This is even more important in the washing step, where we're trying to get very, very high levels of impurity removal. Single stage washing would require simply unacceptable amounts of wash buffer. But as we go to 2, 3, or even 4 stages, we can get 10,000-fold reductions in the impurity concentrations while still maintaining reasonable levels of buffer usage for this type of chromatography process. Now, in order to make this work, we need to be able to operate the membrane units continuously for an extended period of time. In order to do this, we operate these membrane units below what's known as their critical filtrate flux. This is typical data from a flux stepping experiment that's used to evaluate the critical flux. We stepwise increase the flux and look at what happens to the transmembrane pressure. At low values of the flux, the pressure remains quite stable. But as we increase the flux, we eventually get to a point where the pressure first becomes unstable. And this is what we define as the critical flux. And we choose to operate usually about 10 to 20% below that critical flux to avoid membrane fouling in the system. This is typical data for the pressure profile in the binding step, operated below the critical flux. This was for an 8-hour process. You can see the pressure remains completely stable. There's no fouling of the membrane modules. In addition, the maximum pressure is only about 2 psi, in contrast to the 100 plus psi that's used in column chromatography. This makes it very easy to develop fully disposable systems in which the entire flow path is just plastic tubing without any of the stainless steel piping that might be required for large pressure operation. As I mentioned, this process is continuous. This is the profile for the eluted product. We start with an initial equilibration of the system where we're not generating any product. Just before the 2-hour point, we add in our monoclonal antibody feed, and you can see this is the absorbance profile coming off of the elution stage. It is completely steady state. It's a short residence time process, meaning that there's less opportunity for protein degradation or aggregation, and it allows us to use this as part of a fully continuous downstream process. This is a comparison of the performance of the system used for antibody purification, specifically looking at the host cell protein level, as well as the aggregate concentration. In our clarified harvest for this particular feed stream, we had 54,000 parts per million of host cell proteins as determined by an ELISA. We were able to reduce that to 210 parts per million in our CCTC system, which is actually threefold better than what one can do in a packed column operating under the same conditions. We think that was because we actually designed a better washing process for our continuous process, and we actually had slightly lower levels of aggregate. More significantly, the productivity of this continuous process was more than 4 times greater than that for a column system because all of the resin was in use at all times. There are numerous advantages of this system. There's no columns in packing, and thus there's reduced labor costs and reduced validation costs. It's truly continuous operation, allowing us to get higher productivity. And as I mentioned before, it's a fully disposable flow path. In addition, there are a number of things we can take advantage of in this type of continuous chromatography system, which would be impossible in a packed column. Right now, packed columns typically use particles that are at their smallest about 45 microns in diameter. And the reason for this is that if you go with any smaller particles, the pressure drop through the column becomes exorbitantly high and can actually cause damage to the chromatography particles. This constraint, though, of pressure drop is completely eliminated when we use a flowing slurry, because the pressure drop for pumping a slurry of 45 micron sized particles is basically identical to that for using 25 micron particles. And so we worked with Purolite, who is one of the manufacturers of Protein A resins, and designed a smaller prototype particle resin that could still be used for Protein A chromatography. These have an average particle size of just about 20 to 25 microns. Because of the smaller particle size, it is much shorter time required for the antibody to diffuse into the center of the particles to bind to the protein A ligand. And thus we could get much shorter residence times in our binding step compared to what one would achieve with 45 micron particles. And this smaller residence time allows us to develop a higher productivity continuous chromatography system. In this case, we were comparing a column with 45 micron particles to our continuous system with 45 micron particles, and then our continuous system with 25 micron particles. They all provided very similar yields of antibody and similar reduction in host cell proteins, but the big difference is in the productivity. And the productivity of the continuous chromatography system using the 25 micron particles is twice that in our continuous system with 45 micron particles, and about 12 times larger than that in the column. Note that we could not operate our column with 25 micron particles because the pressure drop required to flow through that column would have actually crushed the particles and made it impossible to do the chromatography. I do need to acknowledge some of the collaborators on the continuous chromatography system. This technology is being developed by Chromatan Corporation. Oleg Shinkaj is the CEO and president. We obtained funding from the NIH, the FDA, and the Ben Franklin Technology Partners, and collaborated, as you can see, with a number of companies, in particular Eli Lilly, Merck, Regeneron, Fujifilm Diosynth, and Spectrum Labs, in addition to Purolite. And as a point of disclosure, I currently serve on the board of directors for Chromatan. In the time that I have remaining, I want to go through one more example, which is work that my lab is currently actively pursuing in looking at a precipitation process as a low-cost alternative to protein A chromatography for initial product capture. Now, precipitation has been used in the past for the purification of plasma proteins. And one of the reasons that it works well for commercial purification of plasma proteins is that the proteins are present at very high concentrations within plasma. Now, the original monoclonal antibody processes had relatively low titers of antibodies, making precipitation largely impractical. But today, it is very possible to get antibody processes that give titers of 10 grams per liter or higher coming out of the bioreactor, which is even larger than the IgG concentration that is present in plasma. And thus, we've been looking at the design of targeted precipitation processes that could potentially be used for initial clarification as well, or initial purification, as well as concentration of monoclonal antibodies directly after clarification, leaving a bioreactor system. Now, most of the work that we've done has been using zinc as a selective precipitator for monoclonal antibodies. These actually couple with the histidine residues in antibodies to form transiently or reversibly cross-linked species that will ultimately precipitate. And we can reduce the amount of zinc that we need for this process by combining the zinc with polyethylene glycol, which is a volume exclusion agent that essentially pushes the antibodies closer together and encourages their precipitation. The way we do this in practice is to use a continuous tubular precipitation reactor that employs static mixers to obtain very uniform residence time. We take our harvested and clarified cell culture fluid, mix it with zinc in the first reactor to nucleate precipitate. We then add polyethylene glycol to enhance the precipitation. And then what comes out of this will be precipitated IgG with all of the host cells proteins remaining in solution so that they can simply be filtered away from the monoclonal antibody product. The total residence time in this system is less than about 45 seconds, allowing us to very quickly get our precipitated protein, which can then be purified by a membrane filtration step. This is the yield of monoclonal antibody after the zinc chloride and PEG precipitation. You can see that we get about 98% yield continuously over 24 hours of operation. The way we would run this process as a full process is we would do our two-stage precipitation. We would then dewater in a membrane unit, which would concentrate the precipitate as well as removing some of the host cell proteins. We would then go through a staged washing procedure, very similar to the countercurrent staging that I described in both the formulation process for diafiltration as well as the countercurrent tangential chromatography. We would then redissolve the antibody by adjusting the pH using a glycine buffer. This would also drop the pH, which would allow us to go directly into a continuous virus inactivation step and then into the rest of the downstream process. This is early work showing a size exclusion chromatograph of the purified monoclonal antibody product. You can see the antibody is the large peak that's present in the SEC. There is a small amount of host cell protein, which are still present in the system. We think these low molecular weight species could still be removed, but it would require increasing or enhancing the washing step that we're using for the purification of the precipitated protein. Well, let me wrap up here. I hope I've been able to show you how one can rethink about the use of or the development of processes for continuous purification of monoclonal antibodies as well as other biopharmaceutical products. Continuous processing as a strategy has the potential to dramatically reduce the cost and improve the flexibility of biomanufacturing processes. It does require a new perspective on the downstream process, and much of this provides new opportunities for the design and development of novel membrane systems. The idea of using countercurrent contacting or staging is a general approach that can allow us to convert batch membrane systems into continuous membrane systems. And I've given you three examples, one for formulation using a countercurrent stage diafiltration, one for purification using a countercurrent tangential chromatography system, and one for antibody capture based on precipitation in combination with a membrane filtration step. Finally, I need to acknowledge the graduate students in my group who actually did all the hard work on the projects that I described. Chris Yale and Mario Jabra led the efforts on the continuous countercurrent diafiltration system. Chris finished up this summer and is now at Spark Therapeutic. Mario Jabra is working at Sanofi. Zhao Li led the work on the continuous precipitation and is now at Merck. The precipitation studies have been done in collaboration with Todd Probitzian and his group at RPI. And we've received funding from the American Chemical Society Pharmaceutical Roundtable, the National Science Foundation, Membrane Science, Engineering, and Technology Center, which is part of their Industry University Cooperative Research Center program, and an NSF Goalie Award that Todd Probitzian and I were the co-PIs on that allowed us to get the precipitation studies started. With that, I will stop, and I will be more than happy to answer any questions that you might have.
Thank you, Dr. Zydney, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. First question is, did you check another ion for selective precipitation?
That's a great question, and there would be other divalent cations that could be used as an alternative to zinc chloride. There's some literature on the use of calcium that has shown reasonable performance. What we have found is that the best precipitation in our lab has been obtained using the zinc chloride system. Zinc is also well established as a precipitating agent in the production of insulin, and so we have focused most of our efforts on looking at the zinc polyethylene glycol system. But the technology would not be limited to that as a precipitating agent.
Well, thank you, Dr. Zydney. Do you have any final comments for our audience?
No, I hope you found the webinar useful. Please don't hesitate to email me if you think of a question afterwards, and I would be happy to follow up. And I hope you enjoy the rest of the bioprocessing event.
Well, thank you again, Dr. Zydney, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 6th of 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.